Hello everyone, welcome to my channel Anupma Biology Classes. In this video, our topic is plant growth. This video is taken from Plant Physiology series, CBSE class 11th, part 1st of chapter 16. Now, let's see here the lecture overview. So in this video, we will focus on some important topics related to growth like growth in plants. Plant growth generally is indeterminate, measurement of growth, phases of growth, growth rates and conditions for growth. Now, let's see here the first topic, which is growth. So, my question is, what is growth? Unlike many animals, plants typically continue to grow throughout their lives. The fertilized egg, that is known as zygote, for example, when grows, changes from a single cell to a multicellular plant body composed of roots, leaves, and stems. At maturity, organs concerned with reproduction, that is, the flowers, fruits, and seeds, are formed and grow. These changes occur due to development in plants. So, development is therefore applied to the whole series of changes which an organism goes through during its life cycle. A permanent and irreversible change in the size of a cell, organ, or whole organism, usually accompanied by an increase in dry weight. The development is nearly always associated with the morphogenesis and differentiation. So, what is morphogenesis? Morphogenesis is development of the shape of cells and organs. Differentiation, on the other hand, is applied to the qualitative differences between cells, tissues, and organs. For example, a cambium cell differentiates to produce xylem or phloem cells, or a vegetative bud is transformed into a flowering bud. All the developmental processes involve growth. During growth, the new cells are added by cell division. The organism grows in size, increases in weight, protoplasm multiplicates, and an irreversible permanent increase in the volume takes place. The term growth, so you know that a permanent and irreversible change in the size of a cell, organ, or whole organism, usually accompanied by an increase in dry weight, is known as growth. Though the definition given for the most complex process of growth is more nearly satisfactory, it does not account for the known facts about growth. Actually, no satisfactory definition for growth has yet been coined. Now, here is plant growth is indeterminate or determinate. So see here, growth in plants is unlimited and occurs continuously throughout their life by the activity of meristematic cells. The new cells are continually produced by the cell division in a meristem. So such kind of growth wherein new cells are continually added to the plant body by the activity of the meristem is called the open form of growth. In case of plants, there is meristem at the apex of every root and shoot, known as root apex and shoot apex respectively. So, some plants have intercalary meristem. Here in this picture you can see the intercalary meristem. And grasses, for instance, have such meristem at the bases of internodes. The vascular cambium is another kind of meristem which develops secondary meristem. The root apex and shoot apex add new cells to cause linear growth parallel to the long axis of the plant body, and it is called primary growth of the plant. The secondary vascular cambium and cork cambium add new cells to cause this increase in girth. It is called secondary growth, which occurs mainly in gymnosperm and dicotyledons. So here you can see that the primary growth and the second is secondary growth, where the secondary vascular cambium and cork cambium form. The daughter cells derived from cell divisions in the meristems enlarges and bring about visible increase in size of the plant. The expanding cells differentiate to form the different tissues and become part of the mature organ. So in this picture, you can see that diagrammatic representation of the sites of the zootypical meristem and root apical meristem and vascular cambium. Direction of growth is shown with the help of arrows. Now, here you can see that what is the differences between the determinate growth and indeterminate growth in case of plant. So first one is indeterminate growth. The botanical definitions of determinate and indeterminate tell us the science behind basic growth patterns. Indeterminate growth does not stop, also called weaning tomatoes. The main stem will just keep on growing. Indeterminate growth can also refer to sequential flowering that starts at the bottom and on the sides of a plant and then moves in and up, can reach heights of up to 10 feet, although 6 feet is considered the norm. They require substantial caging and or staking for support, pruning and the removal of suckers is practiced by many but is not mandatory. Indeterminate varieties are not usually recommended as container plants. Now, see here the determinate growth. Determinate growth is finite, also called bush, includes varieties that are bred to grow to a compact height, approx 4 feet. It usually means the main stem ends with a flower or other reproductive structure. Flowering among determinate plant varieties start from the middle and the top and moves downward and outward. They may require a limited amount of caging and or staking for support, should not be pruned or suckered as it severely reduces the crop and will perform relatively well in a container, minimum size of 5 to 6 gallon. So this is the difference in the determinate and indeterminate growth. Now compare and contrast between the primary and secondary growth. So in case of primary growth, growth in length occur, but in case of secondary, growth in girth occur. In case of primary, apical or intercalary meristem, that is protoderm, procambium and ground meristem occur. While in case of secondary growth, lateral or secondary meristems, that is the vascular cambium and cork cambium. Meristems are clusters of undifferentiated cells in case of primary growth, while the cylindrical meristems in case of the secondary growth. Primary growth occurs in the region of the new growth of the plant, while the secondary growth occurs in the region of a woody plant where primary growth has ceased. So these are the compare between the primary and secondary growth. Now let's see here the measurement of growth. So how can we measure a growth of a plant? Growth in plants can be measured in terms of increase in the length or girth, that is in case of stem and root, increase in fresh or dry weight, increase in area or volume in case of fruits and leaves, or increase in the number of cells produced. Practically, an individual measurement is sufficient, but more information will be required to get correct result. For example, when a seed is germinated and allowed to grow in dark, it elongates rapidly. At the same time, the dry weight of the seedlings decreases because it utilizes the reserved food stuff to grow. A dry weight measurement in such conditions will be misleading. Growth as measured by the total fresh weight of plant can also be misleading as the water content of a plant changes frequently with a change in environment. Measurement of length alone need not to give correct result in many cases. Thus, specific parameters are employed to measure the growth of different organs are, for example, first is increase in fresh weight, increase in dry weight, increase in length in case of pollen tube, increase in volume, increase in cell number, increase in surface area in case of leaves. So while measuring the growth of the plant or plant organs, one should select the kind of measurement which is mentioned here, that is increase in fresh weight, increase in dry weight, increase in length, increase in volume, increase in cell number or increase in surface area. More than one parameters may be employed to get correct results. Generally, the growth of a particular organ is measured by a particular parameter. For example, growth of a dorsiventral leaf is measured by measuring the increase in surface area. Growth of shoot is measured by measuring the length as the shoot apex generally grows by increasing the length, and measured by measuring the volume as the fruit generally grow by increasing cell volume. So the growth of pollen tube is measured in terms of its length. Now, after this, see here the phases of growth. So what are the phases? There are three phases of growth. First one is cell division, second is cell enlargement, and the last is cell maturation. We will see each and every part in detail. So at first, cell division. A cell is metabolically highly active at the time of cell division. Its cellular mass increases and the replication of genetic material, that is nucleic acid, takes place. Growth as a result of division is based on mitotic cell divisions. In the stage of mitosis, each chromosome splits lengthwise into two homologous chromatids which pass equally into daughter cells. Certain cell organelles via chloroplast and mitochondria apparently duplicate themselves. As a result of division, each of the two daughter cells is only half the size of the parent cell. These cells then proceed to enlarge and synthesize their own cytoplasm and cell wall materials. Now, cell enlargement. Cell division is followed by the cell enlargement before actually the maturation takes place. The cells increases in size mainly due to vacuolation, that is by absorption of water. A big central vacuole appears which pushes cytoplasm to be limited to a thin boundary layer against the cell wall. The new cell wall material is synthesized to cope with the enlargement. So here you can see that increase in that means cell enlargement, cell increases in size. Now the last is cell maturation. Cellular differentiation, which is followed by cell division and enlargement, leads to the development of specialized mature tissues. For example, the meristematic cells of leaf epidermis differentiate to form guard cells, trichomes, glands, or epidermal cells. The differentiation and maturation of various kinds of cells depend on the number of internal, external, nutritional, genetic, and hormonal factors. Now, in case of plant root zones, first one is meristematic zone, where the cell divides both in direction of the root basis to form cells that will become the functional root, and in the direction of the root apex to form the root cap. In root zones, second is the elongation zone, where the cells elongate rapidly, undergo final round of divisions to form the endodermis. Some cells thicken to form Casparian strip. The last is maturation zone, where the fully formed root with the xylem and phloem root hairs first appear here. So these are the zones or phases of the growth. Now, after this, demonstration of phases of growth. So see here, to demonstrate and study the phases of growth, germinate a few sets of pea or bean on a circular filter paper soaked with the water in a petri dish. After about 2 days of growth, select a few seedlings with a straight radical of 2 to 3 cm length. Blot the surface of radical with a blotting paper and mark the radical from tip to base with at least 10 points at intervals of the 2 mm using waterproof ink. As soon as the ink dries, put the seedlings again on a moist filter paper in a petri dish and allow the seedlings to grow for about another 2 days. Now measure the length of radical by placing it on a graph paper that is placed below the glass sheet. Measure the intervals between the marks. It is observed that the intervals immediately behind the apex have elongated most. This is the zone of elongation. So here you can see that this is a figure of demonstration zones of the elongation in the young root. The portion further away from the apex, that is the more proximal to the phase of elongation, is the phase of maturation. The cells of this zone have attained their maximum size in terms of wall thickening and protoplasmic modifications. So this is the demonstration of phases of growth. Here you can see that the seeds grow in size, that means in germination, then seedlings occur, then the mature plant. So this is the life cycle of a plant. Now after this, see here the growth rate in plants. So what are the growth rates in plants? Growth of an organism or part of the organism is expressed in the form of growth rate, which is defined as increase of growth per unit time. For example, increase in the number of cells per unit time or increase in the weight of the organism per unit time, etc. Thus, the growth rate of an organism may be expressed arithmetically or geometrically. So, the increased growth per unit time is called growth rate. The growth rate can be arithmetic and geometrical. So, in case of arithmetic growth rate, only one daughter cell continues to divide, which you can see in the picture, and another daughter cell differentiates and matures. That is elongation of root. Example is. And in case of geometric growth, initial growth is slow, that is lag phase, followed by a rapid increase in growth, that is log or exponential phase, and followed by a primary where growth slows down, that is stationary phase. Example like the cells, tissues and organs. So let's see here in detail. First is in case of arithmetic growth, as in root apex and shoot apex, single meristematic cell divides by mitotic division to form two daughter cells. Now, one of these daughter cells differentiates to become a permanent cell, whereas the other one remains meristematic and divides again by mitotic division. The process continues and new cells are constantly added to the body, resulting in the constant elongation growth. If the length of the growing organ or the organism is plotted on a graph against time, a linear curve is obtained, which you can see in the picture. So arithmetic graph is linear curve, and mathematically it is expressed as LT equal to L0 plus RT, where LT equal to length of the root at a time t, L0 equal to length of the root at time 0, and RT equal to A by B, that is elongation of root per unit time or growth rate. So, the growth of an entire plant or the plant organs characteristically passes through different phases. The time course of the growth plotted on a graph paper shows S-shaped or sigmoid curve. Here you can see that. It has been shown that the rate of growth is initially slow, known as lag phase. The growth rate then gradually becomes more and more rapid. This phase of growth is termed log phase or exponential phase. Subsequently, the growth rate slows and finally stops. The latter is termed as a steady phase. So, the total period of time during which all the phases occur in is called the grand period of growth, and such type of growth that occurs in many higher plants and plant organs and measured in size or weight is called geometric growth. The exponential growth may be expressed as, here you can see that W1 equal to W0 ERT, where W1 equal to final size of the root, W0 is equal to initial size of the root at the beginning of the period, R equal to growth rate, T equal to time of growth, and E equal to base of natural. That means logarithms. So increase in the amount of bulk of growth, that is increase in quantity, is compared between the growth of living system in the two different ways. So how can we compare here? The first one is absolute growth rate and the second is relative growth rate. So in case of absolute growth rate, increase in total growth, here the first leaf shows, of the two organs or the organism is measured, and comparison of the total growth per unit time is called absolute growth. While in case of relative growth rate, the growth of the given system per unit time expressed on a common basis is called relative growth rate. So the absolute and relative growth rates can be explained using the growth in size of the two leaves. The absolute growth rate is same, but the relative growth rate is different. So here in this figure you can see that the relative growth rate of leaf B is more as compared to that of leaf A. So this is all about the growth rates. Now let's see here the conditions for growth. So what are the conditions for growth? The plant growth is influenced by a variety of external and internal factors, and these factors include supply of nutrients, water, oxygen, suitable temperature and light. So a brief account of these factors is given below. First one is supply of nutrients. Second is C by N ratio. Third is temperature, then water, and the last is light. So these are the conditions for the growth. All plants need water, nutrients, and oxygen to grow. Normally, plants can spread their roots to take these in from the soil. Potted plants have limited space for their roots to grow. Potting compost helps to hold more water, air, and nutrients. Rooting compost is good for drainage. Various materials in potting compost. Loam based compost have good garden soil. Loam less compost have a lot of peat, and the rooting compost to help cutting to roots and lot of sand perlite. So these are the conditions of growth. First one, that is the supply of nutrients. Plants require inorganic ions for their normal growth and development. For example, carbon and oxygen is in carbon dioxide and hydrogen in water are assimilated in photosynthesis. The carbohydrates thus synthesized provide carbon skeleton which are incorporated into amino acids, proteins, nucleic acids and many other plant constituents. All these metabolic processes require inorganic nutrition. The growth depends directly upon the availability of food material and the supply of essential elements. However, under nutrient deficient conditions, the plant growth is very much affected. If we talk about C/N ratio, the ratio of carbohydrates and nitrogenous compound regulates the specific pattern of growth in plants. For example, if a plant contains more nitrogenous compounds as compared to carbohydrates, it produces more protoplasm. Less mechanical tissues and the vigorous vegetative growth, the shoot is large in comparison to root. The leaves are large, soft, succulent and dark green in color. On the other hand, less nitrogenous compounds and more carbohydrates favor synthesis of the more wall materials, less protoplasm and more mechanical tissues. In case of temperature, plant growth is the outcome of cellular metabolism. Since the metabolic activity of the plant is directly affected by the variations in the temperature, the growth rate is also influenced. The rate of growth in plant tissues increases with increase in temperature to optimum values, after which it declines as the temperature becomes excessive. Fluctuations in day and night temperatures favor a better growth. For example, growth in many plants is best when the day temperature is 25 degrees centigrade and night temperature is 12 degrees centigrade. Fluctuations in temperature influencing growth and metabolism is termed as thermoperiodicity. So, effect of high temperature on plant is C3 plants are more susceptible to high temperature stress than C4 plants. High temperature causes injuries to the chloroplast by altering structural organization of the thylakoid, swelling of grana, and impairing grana stacking availability. Now water. The water mainly absorbed by the roots is used in photosynthesis and then metabolic process and ultimately lost by transpiration. Excessive transpiration or water deficiency in the soil causes water stress in plants. It has been observed that growth is considerably retarded during water stress and sometimes excess water stress stops growth. Next is light. If a plant is allowed to grow in complete darkness like the picture, it shows marked etiolation, that is phenomena exhibited by the green plants when grown in darkness. The hypocotyl elongates profoundly and the cotyledons do not expand if the seedlings is epigeal type. In hypogeal seedlings, it shows greatly elongated internodes, small leaves and the hooked apical bud. The vascular elements are poorly developed and the plant is pale, thin and sickly in appearance. Light grown seedlings, on the other hand, are sturdy, green, with shorter internodes, expanded leaves, and upright apex. As far as the plant are concerned, there are two major effects of light, one related with photosynthesis and the other with the growth and morphogenesis. The control which may be exerted by the light over growth, development and differentiation independent of the photosynthesis is called photomorphogenesis. Most of the process in plants, for example, seeds germination, the growth of seedlings, differentiation of various tissues and organs and the reproduction and controlled by light. So these are the conditions for growth, and this is the end of video in which we discussed about the growth, its measurement, phases of growth, growth rates and conditions of growth. The next upcoming video will be on the plant development. So if you understand this video, please like and share it and subscribe to my channel Anupma Biology Classes. If you have any question, any queries or any suggestions, you can ask in the comment section below. Thank you for the watching.